Let me welcome everyone to this year's Carl and Gerty Cori lecture. This is the most distinguished lectureship that our department provides. And we're very fortunate to have as our speaker today, Tom Rapoport. And I trust that you all have received a little brochure which we normally would hand out, but what's on there is information about the Coris and about Tom. And I am going to just have Carl Frieden tell us a bit about the Coris and the lectureship, and then I'll have Wei-Kai Lee introduce Tom. Carl, do you want to talk about the lectureship?
Well, I wanted to make a few comments about the Coris.
Right, I'm sorry.
I was fortunate enough to meet and interact with both Gerty and Carl Cori. And in my comments today, I thought I would make a few different, talk a little bit differently about my interaction with them. And I wanted to talk about the lunch table. I joined the department as a postdoctoral fellow, but my mentor was not Carl Cori. Rather, I met them at lunch. And the Coris packed their lunch every day and brought it into the medical school. And at lunch, they would sit around a very large table in what was then the biochemistry library. And anybody who wanted to eat lunch with them was entitled to do so, whether they were in his lab or not. And I took advantage of that. And so I was, every day we would come in and eat lunch with both Carl and Gerty Cori, and as did maybe a half a dozen other people. Gerty was actually quite sick at the time, but she came in as often as she could. And she was a voracious reader, and she would partly discuss some paper that she had recently read and what the comments of the people around the table might be. Even after she died, after a couple of years, after I came, Carl continued to have those lunch meetings, and they were both brilliant people. They would be those rare individuals to whom all products of human intellect were accessible. So Carl was equally at home discussing archaeology, music, botany, and any other subject. He was in many ways taciturn, but he was amusing, but he was still intimidating. At the time that I joined the department, glycogen phosphorylase was one of the major interests of the Coris. And they discovered what I considered to be the first actual allosteric enzyme. At least the data were published for that AMP effect on glycogen phosphorylase B. I would think that I don't think that at that time that they recognized that allosteric enzymes, or at least those kinds of enzymes which are affected by non-substrate compounds, were going to become a basic tenet of biochemistry, because the term allosteric was not invented for many years after their initial observation. For their work on the glycogen phosphorylase, and like they shared the Nobel Prize in 1947, and Gerty was the first American woman to do so. Their lab attracted a huge number of people, 6 of whom went on to win Nobel Prizes of their own. All of this is actually discussed in a book I'm showing, which is called The Crucible of Science, which was written by John Exton from Vanderbilt University, and describes in detail, traces the history of the biochemistry through the eyes of the Coris, and those who interacted with that remarkable couple. And that's the end of my comments for today.
Okay, thank you, Carl. Thank you very much. Yeah, the book is available, and we have information on our website about how you can get it. So let me turn it over to Wekai, and let me just check with Alex. If people have questions during Tom's talk or at the end, are we going to have them put them into the chat? Oh, maybe we've lost Alex. Wekai, why don't you take over? Go ahead.
Okay. So really glad that Tom is here with us today. Tom received his PhD from Humboldt University in East Germany. He then became a professor at the Max Planck Center for Molecular Medicine in Berlin before he came to US. Tom joined Harvard Medical School in 1995, and within 2 years, he was elected to the Harvard Hills. He's also a member of the National Academy of Sciences, the American Academy of Arts and Sciences, the German National Academy of Natural Sciences, and an AAS fellow. So in the early days, one of Tom's famous accomplishments was on the mathematical modeling of the metabolism. So along with Reinhard Henrich, they developed a metabolic control theory, which pioneered the field now called systems biology. For most of his later career, Tom's research centers around the endoplasmic reticulum, or the ER. Tom's major contribution is figuring out the molecular mechanism of how proteins are transported across the ER membrane in eukaryotes or across the plasma membrane in bacteria, the archaea. This process is called protein translocation. So during the protein synthesis, the ribosome is attached to this membrane channel called the translocon. So the ribosome pushes this nascent polypeptide chain through the translocon, and signal peptide got cleaved and generates this soluble protein in the secretion pathway. For the membrane proteins, they actually exit from side of this translocon to get into the ER membrane. So the translocon is a very smart machinery that allows both of these processes to precisely work out. So the ER is a very crowded place for the newly synthesized proteins, and proteins can misfold. And so the cells have evolved a quality control system in which the misfolded proteins are usually translocated across the ER membrane and back into the cytosol, then get degraded. So this process is called ER-associated protein degradation, or Hrd. So Tom will tell you about this story today. And Tom's contribution to science are too many to be named. So over the years, his lab has also studied ER morphology. That is how the ER form this curvature and form the tubular network. So he also studied how DNA is transported across membrane, and most recently, how the proteins are imported in the peroxisomes. So without further ado, let's welcome Tom again for the Cori lecture. Thanks, Tom. Let me stop this.
Yeah. Well, first, okay, maybe you can.
Yeah, I need to stop this. Cool.
Well, first, thanks, Wekai, for the kind introduction. I really feel very much honored to be asked to give this lecture, and particularly to be named, to be even use, my name in the same sentence as the Cori name, because I mean, for me, the Coris are like the heroes. I'm actually familiar with the Coris from the time when I was a child, because my parents were scientists, and my parents actually knew the Coris, and they were full of admiration for them. And of course, as a student, I was learning about the Cori cycle, about glycogen breakdown, glycogen storage diseases. All this was kind of, basic knowledge, textbook knowledge already at the time. I also have a connection to indirectly to the Coris, because my first work as a graduate student was on enzyme kinetics. And at that time, I encountered the name Carl Frieden. who already was a big name in the field in enzyme kinetics at the time. And finally, I want to mention another trainee of the Cori's, Ernst Helmreich, who became a very prominent German scientist. And in my very, very first meeting, I met Ernst Helmreich, and he was very friendly and supportive, and I kept contact with him until he passed. And so in a way, I feel like I'm a grandchild of the Coris, even though I never met them. Anyway, so I'm gonna share my screen now. So I'm going to talk about the mechanism of ER-associated protein degradation, Hrd. So let me start out what Wei-Kai actually already mentioned. Many proteins in a eukaryotic cell are translocated across the endoplasmic reticulum membrane through the classic SecY channel. Some proteins are transported all the way across and end up in the luminous side, and other proteins are integrated into the ER membrane. And both classes of proteins subsequently undergo quality control, such that only properly folded proteins are moved on along the secretory pathway or stay in ER. If a protein doesn't reach its native folded state, in other words, it's misfolded, it will be retrotranslocated into the cytosol. We call this retrotranslocation because it is the opposite direction of how proteins got into the ER in the first place, which one might call forward translocation. And then once a protein arrives in the cytosol, it is polyubiquitinated and then degraded by the proteasome. And this is the pathway that we call Hrd for ER-associated protein degradation. So Hrd is conserved in all eukaryotic cells. We study it in yeast because it's a little bit simpler. But it is not only involved in the degradation of misfolded proteins, but also in the regulated degradation of folded proteins. For example, the HMG reductase is degraded when you have cholesterol, and that also happens through Hrd. And it is also hijacked by certain toxins and viruses. For example, cholera toxin moves from the plasma membrane into the ER by vesicular trafficking, and then crosses the ER membrane through Hrd into the cytosol. Hrd is medically important. It is associated with a bunch of different diseases, for example, cystic fibrosis. So what I will talk about today is the mechanism of Hrd, and I will address three questions. First, how are misfolded substrates recognized in the lumen? Second, how are selected substrates moved across the ER membrane? And then third, how proteins are extracted from the membrane and moved into the cytosol. So I want to remind you that moving a polypeptide chain across the membrane is actually not a small deal. It is energetically unfavorable. So membranes are normally impermeable to even small molecules, and let alone macromolecules. So in the case of forward translocation, which my lab has been working on for many decades, we actually understand quite well how it works. So in this case, proteins are moved through an aqueous pore that is formed by the Sec61 channel in eukaryotes or the SecY channel in bacteria in archaea. What you see here is a space-filling model of an X-ray structure determined in the presence of a translocating substrate. It was determined by two former trainees in the lab, Long Li and Union Park. And what you see here in blue is the translocating polypeptide chain, and you can appreciate that it is in a pore that extends all the way from the cytosol to the other side of the membrane. The polypeptide chain is an unstructured polypeptide segment, and it moves in an entirely hydrophilic environment. The barrier is maintained by a pore ring of 6 amino acids that form a gasket around the polypeptide chain. So how does it work in Hrd? So before I go into little details, let me first make the point that there are different Hrd branches. Has been worked out in yeast by a former postdoc in the lab, Pedro Carvalho, but it's likely true in mammals because all the components that I will mention are conserved throughout all the eukaryotes. So there are three pathways, and they are distinguished by where the misfolded segment of the polypeptide chain is located. If it's in the lumen, we call it Hrd1. If it's inside the membrane, we call it Hrd1. And if the misfolded segment is on the membrane protein on the cytosolic side, we call it Hrd1. Hrd1 and M use the same ubiquitin ligase called Hrd1. It's a ring finger ligase, whereas Hrd1 uses a different ligase. So all pathways converge on the cytoplasmic site at the level of an ATPase that is called Cdc48 in yeast and P97 or VCP in mammals. And then from there, they're passed on to the proteasome for degradation. Hrd1 requires two components, not just the Hrd1 ligase, but also an interacting protein that is called Hrd3 on the luminous side. And Hrd1 requires yet additional proteins, in addition to Hrd3, Yos9, Usal, and Der1. So in this talk, I'll be concentrating on this Hrd1 complex and how it allows proteins, either in the lumen or in the membrane, to move out of the membrane and into the cytosol to be degraded. So we know best how it works for misfolded luminal glycoproteins. If a glycoprotein in the lumen of DR lingers around for too long, then the glycan, N-glycan attached to an asparagine, is trimmed back by glycosidases to generate an alpha-1,6 mannose residue called Yos9, which is part of this Hrd1 complex. And then somehow the polypeptide chain makes it across the membrane. Once the segment arrives on the cytosolic side, you get polyubiquitination catalyzed by the Hrd1 protein, the ligase. And then the Cdc48 comes into play and pulls the protein out. So what I'm going to tell you today about is the structure of the Hrd1 retrotranslocation that we determined by cryo-EM analysis of subcomplexes. And all the work was essentially done by a single person in the lab, Xudong, the postdoc who's currently on the job market. So first, I will address the question of how misfolded luminal substrates are recognized and how are they distinguished from the folding intermediates that are very abundant in the lumen of DR. And this will be addressed by a structure that we determined that contains Hrd1, Hrd3, and Yos9. So here we're concentrating on the luminous side of the ER membrane, where the Hrd3 protein and the Yos9 proteins are located. So this shows the structure. And what you can appreciate is that Yos9 has two domains. This is the lectin domain, and where the star is located is where the alpha-1,6 mannose will be recognized and bound. And next to it is a groove that is formed by Hrd3 and in part by the MRH domain. If you look from the membrane, you can appreciate this groove here. And this is where the unfolded polypeptide segment will bind. The groove is too small to allow any folded segment to be bound here. And so although we don't have a structure with a substrate bound really, we can basically already tell that there will be dual recognition. The first recognition is through the glycan, the alpha-1,6 mannose residue that will bind to the MRH domain. And the unfolded polypeptide segment that is adjacent to the glycan attachment site will be accommodated in the groove of Hrd3. And so this dual recognition makes sure that only the properly misfolded proteins that are hopelessly misfolded will be degraded, and folding intermediates that are on the way to become a native protein will be spared. So next I will talk about how selected ER L substrates move through the ER membrane. And we can get insight from this by solving a structure of a Hrd1, Usal1, Der1, and Hrd3 complex. Now, in order to get the cryo-EM structure, we had to use a trick. We had to fuse the Hrd1 protein with the Usal1 protein. So basically, we chopped off all these segments that are shown here with dotted lines and fused the C terminus of Hrd1 to the N terminus of Usal1 to generate a single polypeptide chain. However, it turns out this fusion protein is fully active in Hrd1 in vivo, even when expressed at endogenous levels. So what you see here is a cyclohexamide chase experiment, and we're using a model substrate that is misfolded. And as you can see, in the absence of Usal1 and Hrd1, this protein is completely stable. But in Y-type cells, it is degraded, and the Hrd1 Usal1 fusion is just as active as the Y-type. So this shows the cryo-EM structure of this complex. Here's the density map, and on this side is the model. So here's Hrd3, Hrd1, Der1, and Usal1. I should mention that we don't see much of the cytosolic segments. They are presumably too flexible, so we don't see them. So what I will do now is I will go through the individual components and tell you what they're doing. So first, let's talk about Hrd1. So Hrd1 is a multi-spanning membrane protein. It's the ubiquitin ligase again. So it has 8 transmembrane segments, a ring finger that we don't see in the structure, and a segment that follows it. So the most interesting aspect of the Hrd1 structure is that it has a very large cytosolic cavity that is filled with hydrophilic residues. These are shown here in blue. And this cavity is open towards the lipid interior. So we call it a lateral gate that is between transmembrane segment 3 and 8. So here you can see, here's the cavity, and it would be open towards sideways towards the lipid. So next is Der1. So Der1 was predicted to be an enzymatically inactive member of the rhomboid family of proteases. And indeed, when we compare the structure of the Der1 protein with a rhomboid protein called ClpG, you can see that in an overlay, the two proteins are extremely similar, both in the top view and in the side view. Both proteins have a lateral gate that is located between TM2, transmembrane segment 2, and transmembrane segment 5. In our structure, the lateral gate is closed, but we have another structure in which the lateral gate is actually open. So one difference between the rhomboid and the Der1 protein is this loop here. In the rhomboid, this loop covers a luminal cavity that is shown here. So in the rhomboid, there would be a loop above it. But in Der1, this loop is much smaller, much shorter. And so this cavity is constitutively open. So what can we learn from the rhomboid proteases that have been studied quite well over the years by other labs? So what you see here is the rhomboid protein in its closed state. The first thing that happens is that this loop that I mentioned, which was shown in red in the previous slide, moves away to open this cavity. Then this substrate, this is a transmembrane substrate, moves sideways through the lateral gate into the interior of the protein. A segment of this transmembrane segment gets unfolded and can reach the active site, and then you get cleavage and a segment is released. So in the case of Der1, as I mentioned, it's an inactive member of the family, so it doesn't cleave anything. And we think it functions in reverse compared to rhomboid proteases. So here you have a soluble substrate in the lumen, and then it would insert into this luminal cavity. And then one branch, one half of this hairpin structure would move through the lateral gate between TM2 and 5 into the lipid. So let's have now a look at the Der1 Hrd1 interface. So it turns out the interface between Hrd1 and Der1 is actually very small. These two proteins don't interact very well with one another. And so, in fact, in order to keep these proteins together, you need the Usal1 protein, which serves as a linker. So the Usal1 has a C-terminal region that interacts with Der1, and then it interacts through a flexible segment that we don't see in the structure with the Hrd1 protein. Now, this interface is very small, and so you might argue, well, is it real? Are we sure about this interface? And so in order to test that, we use photo cross-linking. And this is based on an approach that had been developed by Peter Schultz. So just to remind you, I think many people are familiar with this technique. So you express in cells a suppressor tRNA together with a modified tRNA synthetase, and you add into the medium a photoreactive amino acid derivative. And so this modified tRNA will charge this tRNA with this amino acid, and then you will get incorporation of this photoreactive amino acid at all positions where you have an EMBER codon. So the EMBER codon will be suppressed by the tRNA, suppressor tRNA, and you get incorporation of the photoreactive probe at a specified position. And then when you shine light, UV light, you get cross-linking between the protein and anything that is in the neighborhood. And so using this approach, we could show that all these residues shown here in red cross-link to the Der1 protein, and another group, the group of Thomas Sommer, did it in the opposite direction. So they put cross-linking probes into the Der1 protein. And as you can see, the green probes all cross-link to Hrd1, the yellow ones cross-link to Hrd3. So these cross-linking experiments very nicely support our structural model. So now let's look again at the interface between Der1 and Hrd1, but this time we're going to look from the ER lumen. And what is really interesting is that the two lateral gates of the Hrd1 and Der1 protein are juxtaposed to one another. So here's the lateral gate between TM3 and 8 of the Hrd1 protein, and here the lateral gate of Der1 between TM2 and 5. And they're next to each other. And both lateral gates are actually accessible to lipids from the side. If you look from the side, in the side view, you can appreciate that in the area where the two lateral gates are facing one another, the membrane is significantly thinner than it would normally be. And this is caused by the luminal cavity of Der1 and the cytosolic cavity of Hrd1. This is a cut through the space filling model, and you can appreciate that here's the luminal cavity, here's the cytosolic cavity. There's only very little space, membrane space left in between. And this is also borne out if we look at the micelle shape in our cryo-EM structure. You can see that the micelle is much thinner here between the lateral gates than it would normally be. And also, if we do molecular dynamic simulations, we can see that the phospholipid head groups shown here in green balls are moving into the membrane and come close to the other side of the membrane. So there's a local thinning of the membrane between the two lateral gates. This local membrane thinning is caused by features of both Hrd1 and Der1. In the case of Hrd1, it is the cytosolic cavity and a lateral opening towards the lipid. And in the case of Der1, it's hydrophilic residues on the cytosolic end of TM2 and also a short TM5. So these are the lateral gate helices. If you look at the TM2 of Der1, you can see these hydrophilic residues on the cytoplasmic end. And if we mutate those hydrophilic residues to make these residues more hydrophobic, we can see that this results in a defect of Hrd. If we mutate them to other hydrophilic residues, there's no effect. Okay, so unfortunately, we don't have a structure of the Hrd1 retrotranslocation with a polypeptide chain yet, but we can again use photocross-linking to probe how a polypeptide chain would move through the Hrd1 retrotranslocation. So a former postdoc in the lab, Pedro Carvalho, did a systematic photocrossing experiment. So he took a model substrate and put the photoreactive probes throughout the polypeptide chain in many different positions. And so if we label the glycan attachment site, the asparagine, at position 0, what he found is that you first, if you go downstream, you get first cross-linking to Hrd3, then to Der1, and then to Hrd1. And these cross-links very nicely fit with our structural model. So in green is a hypothetical polypeptide, the translocating polypeptide. Position 0 would be where the glycan is, the alpha-1,6 monos that would interact with the MRH domain. Position 7 would be in the groove of Hrd3. These two positions would be in the lateral gate of Der1, and all these positions would be in the lateral gate of Hrd1 and its cytosolic cavity. One can also do the experiment in the opposite direction by putting the probes into Der1 and Hrd1. And so we did that for Hrd1, and the Sommer group did it for Der1. And again, these are all the red and green residues are cross-links to where you see cross-links to the substrate, and it fits very nicely with this loop insertion that we already postulated by doing the cross-link experiment with probes in the substrate. So basically what this means is that the substrate inserts as a loop into the Der1 Hrd1 complex. So once this has happened, the polypeptide chain can presumably slide back and forth, but when a suitable lysine arrives on the cytosolic site, you get polyubiquitination, and then the Cdc48 comes in, pulls it out, and delivers it to the proteasome. So I want to compare here forward and retrotranslocation. In both cases, translocation is initiated by loop insertion. So if we first look at the conventional SecY channel or SecY channel, you can see that one part of this hairpin of this loop inserted polypeptide is hydrophobic, either a single sequence or transmembrane segment, and it would move sideways out of the channel into the lipid phase and would then stay stationary. The other part of the hairpin in green here is hydrophilic, and that would move through the entirely hydrophilic pore to the other side of the membrane. Now, this is different in the retrotranslocation situation. Here is the substrate, and now it also inserts as a loop, but in this case, both parts of the hairpin are hydrophilic. And so you need to stabilize both parts of the hairpin. One part would be stabilized by the Der1 protein, the other part of the hairpin would be stabilized by the Hrd1 protein. And the part in between would move through the thinned membrane area across the membrane. And this thinning would reduce significantly the energy barrier to move a polypeptide chain across. So what about Hrd-M? I mentioned at the beginning that Hrd-M only requires Hrd1 and Hrd3, not the other components. So we actually determined a structure of the Hrd1, Hrd3 complex in the absence of the other components. And what we can see is that even when on its own, the Hrd1 protein can thin the membrane locally where the cavity, the lateral gate is located. On the other side of the complex, the micelle just looks normal. And again, using molecular dynamic simulation, we can see that phospholipid head groups invaginate the membrane deeply where the lateral gate is located. So what this presumably means for Hrd-M is that when you have a segment of a substrate with a lesion, let's say you have a hydrophilic residue inside this transmembrane segment, it doesn't feel very comfortable in the normal membrane. So it would have a tendency to go to the distorted thin membrane area, and then it will leave the membrane through the cytosolic cavity of the Hrd1 protein into the cytosol. So the important point here is that there's membrane thinning that is allowing proteins to cross the membrane. There's no hydrophilic channel. So we have this hydrophilic a luminal cavity, which is pretty shallow, but then a deep cytosolic cavity on the cytosolic side by the Hrd1 protein. So we think this translocation through a locally thinned membrane is actually also true in other systems, other translocation systems. I give you a few examples here. Many of these actually came out this year, so actually it's kind of a boom here of different structures that support this idea. So the first example is the YTC protein, which is a bacteria protein that also has homologs in mitochondria and chloroplasts. And it allows transmembrane segments to insert into the membrane. So it means that some segment has to cross the membrane and flip to the other side. And if you look at the YTC structure, you see that it has a very deep invagination on the cytoplasmic side. And again, there is local membrane thinning where the membrane, the head groups of the phospholipids invaginate into the membrane, come close to the other side. These two protein complexes, GET and EMC, are involved in the integration of tail-anchored membrane proteins. So, and again, this is similar to this here, except that this would be the C-terminus. So the C-terminus of the protein would go across. And if you look at these here, you again have these cytosolic cavities that are quite deep and locally and lead to local thinning of the membrane. I should say actually that these two complexes have components that are related to the YTC protein. Here are two more examples. So this is a protein called TIM22, which is involved in the integration of solute transporters into the inner membrane of mitochondria. And if you look at the structure, you can see that it has cavities, hydrophilic cavities on both sides of the membrane, again leading to significant thinning of the membrane locally. And finally, this is the most amazing system actually, twin arginine system, which allows secretion of folded proteins in bacteria. And the most important component here is a protein called TET-A, which has a relatively short transmembrane segment and also a charged residue inside the membrane, shown here in pink. And this protein forms oligomers, and this leads to a very significant thinning of the membrane and presumably allows the folded proteins to move across. So basically what I'm telling you here is that we have a new paradigm, which is that a protein-induced locally thinned membrane is employed to translocate proteins across membranes. In other words, a hydrophilic channel is not always required as we thought previously. So now in the last part of my talk, I will deal with the question of how proteins are extracted from the membrane and moved into the cytosol. And this has to do with the Cdc48 ATPase. So remember, you have this polyubiquitination going on by the Hrd1 complex, and then the Cdc48 ATPase pulls the protein out of the membrane. So I'm going to address the question of how does it do it. So first I need to tell you that Cdc48 is an essential and conserved ATPase. It's called P97 in mammals, or BCP. And it has functions that are beyond Hrd. So it functions in many other processes. So for example, it can disassemble protein complexes or protein nucleic acid complexes. And the important point is that it can act on even well-folded proteins. And because of that, it often functions upstream of the proteasome. The proteasome cannot degrade folded proteins. In fact, it needs always an unfolded flexible segment, either on the N or C terminus, so that it can start pulling on it. So the question then is, how can Cdc48 deal with a broad range of substrates? And it seems that the only requirement is that the substrate has an attached polyubiquitin chain. So let me give you a little bit of background about Cdc48 P97. So it has an N-tron domain, N domain, and two ATPase domains called D1 and D2. Each of these domains has a Walker A and B motif for ATP binding and hydrolysis. 6 of these polypeptides form a hexameric ring structure, actually a double ring structure. So you have the D1 ATPase ring and the D2 ATPase ring. And then the end domain can either sit coplanar with the D1 ring, or it can move up and sit above the D1 ring. And this depends on the nucleotide state of the D1 ring. If it's in the ADP bound state, then the end domains are on the down conformation. In the ATP bound state, mimicked by ATP gamma S, they are in the up conformation. And just for nomenclature reasons, I will call this the cis side and this the trans side. Cdc48 often functions together with a cofactor that is called UFD1 MPF4, so it's a heterodimer, and I will be abbreviating this by UN. One copy of the UN heterodimer binds to a hexamer of the Cdc48 of P97, and the UN complex binds to the cis side. So the understanding of Cdc48 was hampered by not having a really suitable substrate, and the former MD PhD student in the lab, Nick Bodner, made a real breakthrough by developing a substrate. And so the substrate is simply the following. So we have an unfolded segment, it's a degron, and then it's followed by a fluorescent protein. We use either GFP or another fluorescent protein called EOS. And then we use purified enzymes to attach a single polyubiquitin chain with K48, lysine 48 linkages, which are the linkages that are preferred by the Cdc48 ATPase. We also often attach a fluorescent probe at the N terminus because this is brighter than the GFP or the EOS. So I'm not going to show you any data here, but I would just summarize what Nick found in this scheme. So I'll take you slowly through it. So this is the original, the ground state of the ATPase. The D1 ring is in the ADP bound state and the end domains are on the down conformation. Upon ATP binding to D1, the end domains move into the up conformation. The UN complex can bind to the N-terminal domain, and then the ubiquitinated substrate will bind to the UN complex on the cis side. This binding stimulates ADP hydrolysis in the D2 ring, and this results in the polypeptide chain being pulled through the double ring to the other side of the membrane from the cis to the trans side. And this results in the unfolding of the substrate, which we can measure by the loss of fluorescence of the protein. And then once the substrate is all the way through, in order to release it from the Cdc48 complex, you need a deubiquitinating enzyme. It's called O2-1, which binds to the end domain. You need the end domains to be in the down conformation for O2-1 to function. And then it will cleave off the ubiquitins on the cis side, and the polypeptide chain will be released on the trans side, and then it can be transferred to the proteasome. So much of what I just told you in schemes was actually supported by photo cross-linking experiments. So what Nick did is he put photoreactive probes at different positions along the central pore of the double ring ATPase, and he could show that all of them gave cross-links to the translocating substrates. And another experiment that proved that the substrate went all the way through is by using a protease called FDSH. He took the protease domain of this protein and fused it to the C terminus of the Cdc48. So he generated an artificial proteasome, if you wish, so that when a substrate comes out on the trans side, it will be immediately degraded by this ATPase. And this is what he found. So this proved that the substrate moves all the way across. So it turns out that only the D2 ring needs to hydrolyze ATP to pull the protein across. And it's also the ring that has a canonical aromatic pore ring residues. pore residues. So there are pore loops that face the central pore, and only the D2 ring has them in a canonical form. So then the question is, how can a polypeptide pass through the D1 ring to reach the D2 ring to actually initiate translocation? So there's no flexible segment. Remember I said the proteasome has it, but this one doesn't need it. And so how do you get a substrate segment across the D1 ring into the D2 ring in the first place? So this was answered by a structure that we determined, the cryo-EM structure of a substrate-engaged Cdc48 complex. So let me briefly explain how we generate this complex. So we have this EOS protein, the fluorescent protein with the attached polyubiquitin chain. There is a streptavidin binding peptide tag at the C-terminus. We bind it to streptavidin beads. Then we add the cofactor complex and wash away the excess. Then we will add the Cdc48 ATPase in different nucleotide states, wash away the excess, and then elute the whole thing with biotin. And this is being then put on the EM grid and analyzed by cryo-EM analysis. And I want to stress that this corresponds to the structure that we have, corresponds to an initiation state of substrate processing, because we use the Walker B mutation in the D2 ring, which means that it was only hydrolyzing ATP very slowly. And we also use very short incubation period with ATP. So this shows the cryo-EM structure. So let me orient you. So here's the D1 ring, the D2 ring. These are the end domains. This is the NPL4 in this brownish color or reddish color. We don't see much of the other component of the cofactor, the UFD1 protein. We see this little bit of piece here, but there's a structural domain that is very flexible that we don't see. So what do we see of the substrate? Well, first, we don't see the EOS protein, the fluorescent protein. What we do see are 3 ubiquitin molecules. There are 2 folded ubiquitin molecules and 1 unfolded ubiquitin molecule, which I will talk about next. So this, here we cut away some of the subunits in the front. And so again, so these are the 2 folded ubiquitin molecules that we call UB1 and UB2. And this is the unfolded ubiquitin molecule. It has its N terminus in the D2 ring. And then this is the lysine K48, which would be linked with the C terminus of the next ubiquitin. We don't see the C terminal segment of the unfolded ubiquitin, presumably because it's unfolded or flexible rather. So if you look at the unfolded ubiquitin, there's only one part that is maintained from the folded ubiquitin. Here's the structure of the folded ubiquitin. It has 1 helix, and that helix is maintained in our structure here. So that is the only thing that is left. Everything else is completely unfolded. This segment here is bound into a groove of the NPL4 protein. And then this N terminus projects through the D1 and into the D2 ring. Now, interestingly enough, the ubiquitin unfolding does not require ATP binding or hydrolysis. We can show this. It works with ADP. We've shown this by site-specific photocross-linking with probes in the D2 ring. So this is a very unexpected result, I have to say, because it seems that the unfolding happens simply by binding to the Cdc48 ATPase. And if you consider the fact that ubiquitin is a very, very stable protein, you can boil it, it is very surprising that by just binding, it will be unfolded. So then how are the polypeptide chains translocated through the central pore? And so the important point here is that the N-terminal segment of the unfolded ubiquitin is located inside the D2 ring. So again, this is the shows here. So this is the N-terminal part of the ubiquitin, and it's located in the D2 ring. It engages the pore loops of the D2 ring. So here are the the loops of the subunits, we actually have 3 different structures. So in 2 of the 3 structures, we have the pore loops at the same position, and they're forming a kind of a staircase arrangement around the substrate. So what we can say from the structure is how the polypeptide chain would be pulled through the Cdc48 ring to the D2 ring. So as I mentioned, we have 4 or 5 loops, depending on the different structures that we have, in which the pore rings engage the substrate. It turns out that these pore loops interact with every other peptide bond. So every second amino acid has an interaction with a pore loop. And so it works like a conveyor belt. So the lowest subunit is, when it's in the ADP bound state, detaches, disengages, and moves all the way to the top of the staircase. And then when ATP is bound, it re-engages with the substrate. And then everything moves down and drags the polypeptide chain with it to the trans side. And this is going to be repeated during ATP hydrolysis. So back to the initiation. So what we are saying here is that there is a universal mechanism for initiation of substrate processing by unfolding a ubiquitin molecule and pulling on it. So this shows a model of how we envision this to happen. And I have to say this is a little bit, or quite a bit, speculative here. So let's assume this is a substrate and this is the polyubiquitin chain. So the ATPase would pick 5 ubiquitin molecules. You know, the ones that I selected here are random, so it could be another 5 that are neighboring. We know that 2 of these ubiquitin molecules are folded and bind to MPF4, and we assume, although we haven't shown this, that 2 additional ubiquitin molecules bind to the invisible domain of UT1 called UT3. This is the ubiquitin molecule that would be unfolded in this scheme. So it would unfold by binding to the groove in MPF4 and by the N-terminus being inserted across both rings. And so next, you would have ADP hydrolysis. You would pull on the N-terminus, and this would dislodge this segment that was bound into the groove, and will also dislodge the ubiquitin molecules bound to the MPF4 tower. Now, it would pull the next ubiquitin into the pore. Now, if the substrate is right next to the unfolded ubiquitin, you would pull the substrate through the central pore next. But if not, you would have to plow through additional ubiquitin molecules and unfold them and pull them to the other side before you reach the substrate. Eventually, the ubiquitin molecules that were on the proximal side of the substrate would end up on the trans side, and we assume that they're refolding on the trans side, whereas the substrate remains unfolded. So you end up with a situation where you have some ubiquitin molecules on the cis side and other molecules on the trans side. And then, as I mentioned before, you need to hydrolyze ATP in the D1 ring. Then O2-1 comes into play, cleaves off the cis ubiquitin molecules, and you release the molecules on the trans side. Now, I want to make the point here that what is released is a heterogeneous mixture of oligoubiquitinated polypeptides. In our experiments, we can see that a large percentage, maybe more than 70% or so, have ubiquitin chains that are longer than 4. And those would be suitable substrates for the proteasome and don't need any extension. There are some molecules that have shorter ubiquitin chains, and they would not be appropriate substrates for the 26S proteasome and would have to be re-ubiquitinated. Okay, so this brings me to the end of my talk. I want to acknowledge the people who did the work. So as I mentioned, Xudong was the major person on the structure of the Hrd1 complex. The molecular dynamic simulations were done by Mark Sigel and Gerty in Frankfurt. We got help from Sergei Ovchinnikov with Rosetta program. And then Wei Mi and Mao Fu Liao, our colleagues, were helpful in early structures. And then the Cdc48 function was done by Nick Bodner. And the structure of the substrate-engaged Cdc48 complex was done by Toomey and Jurgen G with the help of Nick Bodner. And we collaborated on cross-linking on first HD experiments that I didn't have time to talk about, but with Thomas Wales and John Engen, and on cross-linking and mass spec with Scott Ficaro and Jared Marker. And before I close, I'm going to show a very little video here that I received today from Peter Walter. I think it's pretty funny. Okay, thank you very much.
Alex, are you going to field the question?
Yeah.
Thank you very much, Tom. That was great.
Yeah, it's a really, really beautiful talk. So the way we're going to do questions, if people have questions and people are already getting on it, you can either type them into the Q&A or into the chat, and I'm going to unmute you and you can ask your question yourself. So I'm not going to read people's questions out. That we've discovered is not a particularly effective mechanism. So Tao, I'm going to unmute you now, or allow you to be unmuted, and then you can ask your question directly. So, okay. You should be able to unmute yourself now.
Hey, Tom. Can you hear me?
Yes, I can see you, but I can hear you, yes.
Great, thank you for the great talk. So my question, actually I have two questions. The first is, does the channel formed by Hrd1 or Der1 have any kind of protein size limitations or kind of selectivity? I mean, what kind of protein can be?
Right, that's a good question. So we know that glycosylated proteins can move across the membrane, and the N-glycan is very bulky. So this would not work with a narrow channel like in the SecY61 channel, which has a very narrow pore ring. So, so in our case, in the Hrd case, you have, we think the movement of the glycan occurs through the thinned area of the membrane between the two lateral gates. And that, of course, has very little size limitation. So that's why we think that you can move even bulky stuff across the membrane.
Mm-hmm. Okay, thank you. So my second question is, I mean, I'm just curious, does this degradation system have any kind of proofreading function, given some proteins that may be transported there by mistake, or? I'm just curious. This is just...
I don't think we know whether there's any proofreading. I mean, normally, so when you have a misfolded protein in the lumina of the R and you move it into the cytosol, there's really no chance that the protein would refold, right? So it's hopeless. So the protein will be degraded. There are some special cases like cholera-toxin, for example. Cholera-toxin actually refolds once it's moved into the cytosol and becomes an active enzyme. So in that case, it's a refolding, but I don't think that there should be any proofreading on the cytosolic side in general if you have a misfolded protein on the moving across.
Okay, thank you for the answers.
So while we're waiting for some more questions, I can ask one just by taking advantage of my position of power here. You could imagine, I mean, the membrane thinning paradigm, I think, is a really interesting one. And you could imagine sort of two possible limiting scenarios, one in which kind of the membrane composition doesn't really matter that much, or one in which it does. And I tend to imagine that it probably does to some extent. So you know, what is, I guess my first kind of two-part question, one is to what extent does changes in membrane composition vary the ability of to thin and to pull things through? And secondly, I suppose also, are there, if certain types of compositions or local compositions of lipids are more suited than others, do you get local clustering of these things in certain places on the membrane versus others, just because thermodynamically that's the easiest place to pull proteins through?
Right, so both are good questions, and unfortunately, I can't give you a good answer to either one. So what I can tell, first of all, I have to say that our structure is determined in the presence of detergent. So we are looking at a micelle, detergent micelle, not a real membrane. So what I told you is based on the thinning of the micelle and on molecular dynamics with a real membrane. It would be fantastic to have experimental evidence for this. Now, one of the cases that I mentioned, the EMC complex, there is actually a structure determined in nanodiscs, which is a membrane mimic. And in that case, you can also see the membrane thinning. So that's the only evidence that we have that the membrane is actually thin. But in neither case do we have enough resolution to tell whether there's any specific lipid requirement. You would imagine that the acyl chains would maybe shorter acyl chains would be preferred,
right.
But we don't know. What I can tell you is that we have done a reconstitution experiment of a basic Hrd process with just Hrd1. So we can incorporate a substrate into the membrane and Hrd1, and we can observe the movement of the substrate from the lumen of the vesicle to the outside. And in that case, we can actually vary the lipid composition and didn't see any effect. But it's a little bit artifactual because we didn't have all the other components present. Sure. So that's one of the major goals for us, to actually reconstitute the whole Hrd process with the full complex and recapitulate the glycan dependence that we see in vivo.
Got it. Very cool. So there's another question here from Elliot Elson. So Elliot, you should now be able to speak. You should be able to unmute yourself, I should say.
Okay, well, I'm just wondering if the energetics of translocation through a thin region of the membrane.
I'm sorry.
Yeah, especially as compared to translocation through a channel.
I'm sorry, I didn't get the question acoustically. There was some disturbance here.
Has there been an examination of the energetics of translocation through a thin region of the membrane, especially as compared to translocation through a channel?
Right. Yeah, it's a very good question. And I think what we should do is molecular dynamic simulation, kind of steered molecular dynamic simulation with thin membrane versus normal membrane. And I think that would give us an idea of this. Experimentally, it's almost, I don't know how to tackle this kind of question. It's very, very hard to do. Maybe, biophysicists could do it with single molecule experiments or something, but it's a very tough question to address.
Right. Yeah, I think it would be interesting to tackle experimentally.
Yeah.
Maybe by trying to form the membrane.
Molecular dynamics, I think, is possible.
Right. I agree.
We should do it. Yeah.
Yeah. Thank you.
Do we have any more questions for Tom?
I have one, Alex.
Go ahead, Linda.
I was just wondering if your structures gave you any sense of a selectivity for particular ubiquitin linkage?
Well, so we know Cdc48 has a high preference for K48 linkage. So there has been some reports that other linkages are actually helpful. We haven't seen that really, so I'm a little skeptical about these experiments. So it's mostly K48 linkage, I would definitely say.
Did the other linkages actually impede the retrotrans?
Yeah, no, it was, they saw some stimulation of, it was a suggestion that if you have mixed linkages, K48 and K29 linkages, then you would go, that would be better than just K48 linkages. but I don't think it was entirely clear. So this is a paper by Ray Duchesne's lab.
Great. I don't think we have any more questions at this moment.
Okay. Well, let me thank everybody for coming, and let me thank Tom. And we are gonna. I apologize that we can't take Tom to dinner, but he is going to visit with a number of people today and tomorrow, and I want to thank him for making the time.
And I'm looking forward to having this conversations with both the trainees and the faculty.
Great.
Thanks for listening.
Thank you very much.
Thank you.